Okay, welcome everybody to the SSE Presidential Symposium. It is a pleasure to see so many faces, and I am really excited about this lineup of speakers. So we're going to dive right in. Our first speaker is Dr. Brian Donovan. He's a senior research scientist at the BSCS Science Learning, which is, as I learned from reading his CV, one of the oldest, it is the oldest actually, science education organization in the United States. Brian's research explores how genetic education interacts with social cognitive biases and shapes how students make sense of complex biological and social phenomena. He's been a proponent in the past of the idea that the teaching of human genetics is not a socially neutral endeavor. How we teach genetics produces social attitudes about genetic variation and race. And Brian, please lead us off on this symposium.
Thank you, Kelly. Let me just get my screen set up right here. So thank you, everyone, for inviting me out to share my views on genetics education. Today, I'm going to argue that we need to move genetics education beyond teaching about classical forms of inheritance, specifically Mendel, if we want to accomplish this important goal of combating white supremacy. And I'm just realizing that that should be better. And I recognize, that's it's a provocative argument to make, and not just because last year was the 200th anniversary of Mendel's birthday. So I'm going to take some time to unpack what I think about genetics education, its flaws, what I think we need to do about those flaws in today's world. And I'm going to use a bunch of my published and unpublished studies, primarily randomized trials, to make that argument to you. But before I get into that, I want to share a story about my friend Kay. So Kay's a high school biology teacher. She lives in the Midwest. And like many of us here in this room who are biology instructors, most of us have vivid memories of learning biology ourselves, which is why we got into this business. So she remembers sitting in her advanced placement biology classroom, which is kind of advanced biology in the United States, and multiplying fractions and drawing squares when learning genetics, which is something we all have in common. But most importantly, during her learning of genetics, she remembers thinking to herself that her genes were her destiny, that she could not escape her family's history. And this idea that genes are destiny is tacitly, well, directly and indirectly reinforced throughout the genetics curriculum. And several studies show that, not just in the United States, across Europe, and even in South America and African countries as well. So this is a commonplace idea that is present everywhere. And as we know, if we go back through history, this idea of a genetic destiny has been used to justify, racist policies, prejudice, and genocide throughout history. So if we fast forward back to the present, Kay actually ends up earning a PhD in molecular biology at Stanford. We didn't overlap in graduate school. We didn't meet each other there either. We met much later. But, like anyone else who walks into a classroom and is confronted by the complexity and the overwhelming nature of teaching, folks tend to revert back to the way that they were taught themselves. And so she hears herself parroting a lot of different kind of phrases and ideas that DNA is destiny as she's teaching genetics, but deep down, many years of graduate study in molecular biology told Kay that the story of inheritance was far more complex than Mendel and his P's, yet the curriculum that she had and the standards that guided it rarely, if ever, conveyed that kind of complexity to her students. And this discrepancy between what Kay knew to be true, which is the science of inheritance is complex, and what she was telling her students as truth, which is that most forms of human variation boil down to what can be modeled in a Punnett square, that was beginning to gnaw away at her when I met her. And it was at this, we were actually redesigning the high school biology curriculum for BSCS, which is where I work. It's formerly called the Biological Sciences Curriculum Study. We used to publish about 50% of the high school biology textbooks in the United States. And so it was in that kind of context where these worries of hers started to come up as we were thinking about the new genetics chapter. And Kay specifically was worried that, by communicating this idea that DNA is destiny, she was entrenching potentially racist misunderstandings in her students. And that worry wasn't far-fetched given the history of genetics. And so I asked at the end of that meeting, I asked Kate to do something that a lot of people find scary, which is would you be willing to teach your students about race and genetics to counter or challenge flawed genetic ideas and kind of racist arguments? And she said she was up for the challenge, but she understandably had some concerns. And her largest concern was, what if by doing this, I commit a public act of racism and I hurt the Black and the brown students in my classroom? And I said, I completely understand where you're coming from. It's a worry that I've had, that other folks have. But what if doing this kind of teaching didn't harm children? What if it actually helped them? And what if we really need to do this work right now? And you can imagine how she responded to my call to action. And so the same argument that I made to Kay several years ago is the argument that I want to make to you today, which is that if we don't change the way we teach genetics, then we're possibly allowing for the viral spread of misinformation about race and genetics to go unchallenged, essentially, online every day. And this kind of stuff reaches our youth in the United States quite regularly. But if we do change the way we teach genetics, then we have this unique opportunity to pre-bunk a lot of these kind of flawed genetic arguments that go online about race and genetics. And we can actually vaccinate kind of our youth against one particular form of racial prejudice, which we'll get into in a little bit. And just to qualify my argument a little bit, like I'm not saying we should stop teaching about Mendel or classical genetics. I'm just saying we need to move beyond it. And I'm also not claiming that genetics education is the cause or the solution to racism. But I do think that genetics education is this very important cultural venue within the United States where we can do something about racially biased ideas that are present everywhere in our country. And many people might not know that a lot of misinformation about race and genetics is currently spread online. So there are about 15 million high school students in the United States, and 75% of them go online several or more times a day, especially on YouTube. And I have a 13-year-old son, and he does that all the time. So I've ground truth that finding. The problem is that we now know that exposure to misinformation about race and kind of racist messaging is significantly correlated with YouTube usage. And work by Aaron Panofsky at UCLA in particular showed that a lot of this stuff is coming from the white supremacist community. So the white supremacist community online is very effective and systematic in disseminating racist arguments and ideas online in an attempt to kind of draw people into the cause and to reach young men in particular. And a lot of those ideas are coming from racist manifestos. So manifestos written by people like Anders Breivik, who murdered 77 people in Norway in 2011 in a racially motivated hate crime. So these are the kinds of things that you can find online. So this is from Breivik's manifesto. It says blue eyes and blonde hair, many psychological traits, almost never survives a race mixing process due to the fact that Nordic genotypes are recessive, unlike African, Arab, or Oriental genotypes, which are considerably more genetically dominant. So if you just read between the lines here, right, you see a lot of concepts and phenomenon that tend to be discussed during classical genetics. So we have blue eyes, recessive and dominant genotypes. And really, this argument is teed up to resonate with how American youth think about genetics, because remember, the genetics curriculum is largely defined by single gene inheritance and basically Mendel's laws of independent assortment. So this really is like going to impact kids if they come into contact with it. So 17% of kids who go online could be exposed to misinformation like that. And if we combine those numbers, then that's about 13% of high schoolers, or about 2 million high school students could be exposed to misinformation about race and genetics like that. And to put that in context, that's roughly 30 students per middle or high school in the United States. But 98% of high school students take biology at some point during the course of their 4 years of high school. That means really genetics education could be the one place where we have a chance to systematically pre-bunk that kind of thinking and inoculate students against this kind of use of genetic information to justify prejudice stances. And so you might be wondering, am I arguing that, well, how are we going to do this? We're just going to simply teach kids genetics and they're going to be able to sniff out racial misinformation online. And the world is rarely that simple. I think there's 3 barriers that we need to overcome if we want to accomplish this goal. The first is that we need to teach more complicated ideas about inheritance. And currently, the genetics curriculum is not set up to teach about those ideas. And not only that, but genetics education isn't really set up to tackle weighty social concepts like race. And then even if you were to solve those 2 issues, you still have this third problem, which is who's going to be courageous enough to do this kind of teaching in their classroom? Who's going to be like, hey. So, luckily, normally when I give this talk, like I'm not surrounded by a bunch of evolutionary biologists and geneticists and genomicists all the time. So I'm not going to really unpack the biology here, except to say that there are 5 ideas that I think are really important for students to understand. And they're old ideas, right? So the first is, 0.1% of the genome is variable DNA that differs among individual humans. Of course, that doesn't seem like a lot to a novice, but there's 3 billion nucleotides. So we're talking about 3 to 5 nucleotides of difference. That's where the debate tends to be online about race and genetics. Second finding goes back to Lewontin 72, more genetic variation within groups and between groups. So 2 people from 2 different groups are slightly more different than 2 people of the same group, genetically speaking, about 5% more on average. Just like that might be common sense to a lot of folks here, but learning that is essentially learning an interclass correlation coefficient in like an HLM or a multi-level model. So it's actually really hard for kids to learn. Third idea is like, well, maybe this 5% of DNA, that's where all of the, that's where the gene, the variable loci are that are encoding the traits that are causing racial disparities. So, the counter to that is that 2 decades of genome-wide association studies have shown us that complex human traits typically have a relatively weak association with polygenic variation. And of course, it's a correlation that depends on environmental context as well, and a lot of other population factors that differ across groups. And then the fourth idea is super old, which is gene-environment covariance. Because people inherit their genes with their environment, population geneticists tend to be skeptical of the claim that racial disparities are genetic. That evidence of genetic difference is evidence of a genetic cause of racial disparities is something that folks tend to be skeptical of in the population genetics community. And that's because of this 5th idea, which is that no one's ever done a fair scientific test to determine if inequality is due to genes, right? Like we are not currently using CRISPR to do studies to see if that's the case. And we're not running common garden experiments and randomizing children to different environments to test this claim. So no one's actually done a conclusive causal inference study around that claim, right? So I think if you understand those ideas, if kids understand them, then they can sniff out misinformation about race and genetics. So a lot of white supremacists claim that, people of the same race are racially pure and highly homogeneous, genetically speaking. But Lewontin 72 and every study of the partitioning of human genetic variation shows that's not the case, because most genetic variation is found within groups. Some white supremacists argue that there's more genetic variation between groups than within groups. That's not the case, because even though, depending on how we define race, there is genetic differentiation in the human species, right? It's just not as big as a lot of people might think, right? So that idea is flawed. More importantly, some folks claim online that, somewhere at some point or somewhere in the genome, there is this really critical gene or set of genes that determines racial disparities on some kind of trait. And thus inequality boils down to variation in a single gene. But we know that most of the traits involved in inequality are complex traits. Complex traits have complicated explanations that are environmentally contingent. So we know that that idea is flawed as well. And then finally, I think the last idea that the white supremacist community is like unwilling to acknowledge, and yet is an idea that any kid could understand with a high quality science education, is that correlation is not necessarily causation. So if we have 2 groups and they differ in their trait values and their allele frequencies and their environmental exposures, which is often the case when it comes to race, then it's really hard to say if disparities in that trait are due to genes or the environment. And that's because no one's ever done a fair scientific test to get at that. but we do have control over our social environment. It's limited control. We can make new policies, do kinds of things to change the environment. So if we wanted to run a fair scientific test and we can't randomize, then what we can do is just give everyone the same environment. So eliminate all forms of racism that contribute to unequal environments for different groups. Do that, let's say, for 400 years. And if in 400 years, there are still racial disparities in society, well, then you are in a much stronger position to claim that they are genetic, right? But you're never going to hear a white supremacist tell you that racism should be eliminated. They're going to say it's natural, so there's no point in doing anything about it. And they're going to say that's a fact, right? And so what I think kids need to learn is not the kind of ABCs of genetics. It's the causal and the quantitative complexities of modern population genetics and genomics research. And they need to learn those ideas to develop the ability to kind of identify and critique the flaws in the genetic kind of assumptions that are made in white supremacist arguments. So it's not simple. It's very complicated kind of learning that kids need to do. I spent, I think, 4 minutes talking through that stuff. And in a high school classroom, we're talking 3 to 4 weeks of instruction for kids to learn the story that I just shared with you, which is something you guys probably all know. But, make no mistake about it, like the white supremacist community takes advantage of the public's unfamiliarity with the ideas that I just shared with you. So then you might wonder, well, what is genetics education actually doing about this problem? And there's a really good case to make that in the pre-war curriculum, so prior to 1945, genetics textbooks directly reinforced a genetic explanation for inequality. It directly communicated that kind of stuff to kids. Several studies support that. I have some cool slides if anyone wants to look at them later on in the Q&A showing that. They're not necessarily cool, but they really make that case. Today, that's not what's going on in the genetics curriculum. But there's this big worry that potentially genetics education is unintentionally increasing a cognitive form of prejudice, which is called genetic essentialism. So genetic essentialism is the idea that there's more variation, genetically speaking, between racial groups than within racial groups. People of the same race are highly uniform. They're not very genetically different from one another, and that there's some core set of genes that one group has that another group doesn't have, or let's say a set of alleles that causes racial disparities in society. So it's a really strongly deterministic and anti-population thinking view of human difference, right? And genetic essentialism is a thing that I worry about regularly, because there is a very good chance that the way we are currently teaching about genetics, that it could actually reinforce genetic essentialism. So in high school biology textbooks, and this is probably true in a lot of college biology textbooks, the way that race usually comes up is in reference to the monogenic diseases like sickle cell and cystic fibrosis. So you see things like this. Sickle cell is particularly common among people of African descent, or cystic fibrosis, the most common fatal genetic disorder among Caucasians. Those are things that are said in textbooks, right? Often, the incidence of those diseases in other groups is not discussed, or the underdiagnosis of sickle cell in white populations is not discussed at all. But there's good reasons why these diseases are in the curriculum, especially sickle cell, heterozygote vigor, right? There's all kinds of good reasons that we teach about these things. It's true these diseases occur at a higher prevalence within these groups. The question is, when kids learn about this stuff, do they make incorrect inferences about other kinds of racial disparities in society, boiling down to genes? Because we think about it, right? Like if each race has its own special disease, it implies that there's a uniformity within each group. Because different groups get different diseases, it implies that there's some kind of categorical genetic difference between the groups. And it's not unreasonable for a kid to assume that if two groups differ in disease disparities because of genes, then they probably differ for other reasons because of genes as well. Kids are sense makers. We want them to take genetic ideas and use those ideas and concepts to explain the world around them. We set them up to do that. And so we might be actually setting them up toward this genetic essentialist view unintentionally. So my first research program actually looked into this question, and I started it in graduate school, and I didn't have a lot of money. So I did a lot of underpowered randomized trials. So I ran a bunch of double-blinded randomized control trials. And so, I think there's about 100 people in this room. These experiments would work like half of you would be randomly assigned to learn about, read about genetic diseases and race without any mention of race. The other half of you would learn about the prevalence of different genetic diseases in certain racial groups. So the logic of these studies was like, you knock race out of the curriculum in order to study the impact that it has when it actually is in the curriculum, right? In all 3 studies, and they're all published, you can find them if you want to, got the same result, which is that students in the racial condition were significantly more likely to endorse the idea that races differ in intelligence and academic ability because of genes. And only kids in the racial condition would say things like this. If you ask them to explain why are there racial test score differences in New York City, they would say, well, because Black and Hispanic students have genes that cause them to have a harder time learning. So, that's obviously problematic for a variety of reasons beyond just the nature of the prejudice that is potentially being ingrained here. But to me, it points out that Mendelian genetics may be a part of the problem, right? And so that's one reason why I think we need to move beyond Mendel, and why I think Mendelian genetics education might not necessarily be the right place to challenge genetic essentialist ideas. And instead, what I think we need to offer kids is what I call a humane genetics education. And what that means is it involves teaching kids about the complexity of genetics, population thinking, and multifactorial inheritance in particular, for the explicit purpose of refuting genetic essentialist ideas and arguments. So what that boils down to is helping kids to construct accurate models of genetic variation within and between groups and accurate explanatory models for complex traits. And then kids use those ideas to criticize essentialist and determinist models of human inheritance. So you're learning about the complexity of inheritance to refute genetic essentialism. We've done several randomized trials. I'm going to share the results of a trial that we have under review right now. It's a little over 1,000 students nested within 51 classrooms, 15 teachers in 6 different states in the United States. And kids got 28 days of genetics instruction in 2 different orders. So they either got Mendelian genetics or humane genetics, and it was a crossover trial. So half of the classrooms within each teacher got Mendel first, humane genetics second. The other half of classrooms within each teacher got humane genetics first, Mendel second. We survey before and after each crossover. So it's a cluster randomized crossover trial. The kids are baseline equivalent on everything. And this is a well-powered trial. We're at like 90% power on our confirmatory analyses. Just going to share 3 results with you. So what happened to kids' knowledge of genetic complexity, their belief in genetic essentialism, and the extent to which they explain complex trait variation among individuals using genes versus the environment. For those folks who are interested in measurement and psychometrics, I can talk about that later, but these are all really well-behaving psychometric variables. So want to also give a shout out to my research team. Monica, Molly, Dennis, Bray, Avase, and Jeffrey all helped with this randomized trial in one way or another. It was a really big lift. It unfolded over 3 or 4 years to get this done. So knowledge of genetic complexity. If we look at baseline, the kids are equivalent. Then the kids who learn humane genetics grow significantly more in their knowledge of genetic complexity relative to the kids who are getting Mendel. The 2 groups then are differentiated after the first round of instruction. Second half of kids get humane genetics and they grow more relative to the kids who are now getting Mendelian genetics. At the end of the experiment, the kids have significantly greater knowledge of genetic complexity than they did at the beginning of the study. So their humane genetics instruction increases knowledge of genetic complexity. With belief in genetic essentialism, mirror opposite findings. The groups are undifferentiated and baseline equivalent to start. Humane genetics kids decline more in belief in genetic essentialism than the Mendelian kids. The 2 groups are different after the first round of instruction. The second round of kids get humane genetics. They decline more in belief in genetic essentialism relative to the kids who are now getting Mendel. And at the end of the study, the kids who are getting the humane genetics has driven a decrease in belief in genetic essentialism. Now, if we look at environmental and genetic explanations, the first thing to point out is we have mirror opposite results here. Humane genetics causes a decrease in genetic explanations for complex traits. That does not mean that kids give up on genes for an explanation for complex traits. Just means that they've reduced the kind of magnitude, the effect size really of their beliefs about genes. And we see the opposite pattern with environmental explanations. They increase their environmental explanations for complex traits. There's really important finding here at the end. These are the kids who are getting Mendelian genetics after humane genetics. And you can see that they increase in genetic explanations and decrease in environmental explanations, even when they get to Mendel after learning all this stuff about how the complexity of inheritance refutes essentialism and determinism, right? So right there, that's kind of proof in the pudding for me about why we need to move beyond Mendel. It makes most sense to introduce kids to Mendel, highlight the genomic flaws in Mendelian arguments and the limitations in Mendelian models of inheritance. Where does Mendel work? Where does it doesn't it? Move beyond that into the complexity of race and genomics. That to me is the special sauce here, right? And that kind of argument is consistent. Those findings are consistent with other randomized trials and quasi-experiments that my lab has published. And just recently, my friend, collaborator, Jamie Amamiya, who's going to be an assistant professor of psychology at Occidental soon, we just reproduced these results in another randomized trial with 1,000 undergraduates at UC San Diego. So what I think this all means is that humane genetics is a relatively safe and reliable tool for preventing belief in genetic essentialism. And it's safest or best implemented after students have already learned Mendelian genetics. And that's why I think we need to move beyond Mendel. So we're not throwing Mendel out. We are just putting Mendel kind of in his place, helping kids to understand Mendel, understand the limitations of the Mendelian model, that it doesn't work well for complex traits, unless you're going to get into the binomial approximation to the normal curve and additive genetic variation. Like you could do that at the college level. High school level, we're not necessarily doing that. So if you think that this hypothesis is correct, that humane genetics works for the reasons I've laid out, then the next question is really, well, who's going to do this work? And so that kind of begs the question, how did it go for Kay? Remember, she jumped at the opportunity to do this teaching, even though she was afraid of doing it because she didn't want to commit a public act of racism. And, Kay took a real risk in this kind of teaching, a risk that is different than, say, like teaching about evolution in a classroom of students who happen to be perhaps like evangelical Christians. Like that's also a challenging endeavor too. There's a lot of great research on that. But, in Kay's situation, she actually had students who had family members in the KKK. So, I want to point out, like, doing this kind of teaching is scary for a lot of people and for good reason. It's a real challenge that is different from other kinds of teaching and learning. Nevertheless, when Kay finished doing the teaching, she told me it was a huge rush. And that's the kind of thing you say when you've prepared for a big challenge and overcome it and you feel a sense of relief. And, Kay was prepared for this challenge because she had participated in a 40-hour professional development that we hold every summer to do this kind of work. And we designed that professional development to support teachers that they're ready to kind of tackle the difficult questions that kids might have, develop the subject matter knowledge that they might not have, and learn the kinds of things that they need to do this work really well. And, I think Kay felt prepared. She said that because she learned from the lessons herself as a teacher. She felt, confident about the content she was teaching and about the kinds of questions and concerns that kids might have. She knew how to address those kinds of questions and concerns. And she had a real impact in her own classroom with her own students. So this is an email from a parent that Kay received after she finished the unit. And this parent said, "I thought you might like to know, my son said your class changed the way he perceives the world, and he's not entirely comfortable with that," which is a wonderful thing to hear if you're a teacher.
Just giving you a warning, you've got 1 minute.
Thanks. Yep. As a social scientist, I want to quantify that impact. I want to know what's the number needed to treat, how many kids need to get humane genetics to cause one kid to change their belief in genetic essentialism. And without getting into the details, that number is 14. So in a classroom of 30 students, we would predict that 2 kids would change their belief in genetic essentialism. And to put that number in context, if all 15 million kids in the United States, high school students, got humane genetics next year, then we would predict about 1 million of them would change their belief in genetic essentialism. And that's 52% of the estimated total population of high school students who are exposed to misinformation.
Brian, we're going to have to cut you off here for the next
Online.
speaker to begin.
Oh, okay, I was at 29 minutes, but okay.
Thank you, Brian. That was excellent, amazing. Thank you for sharing that and for launching us talk. I am really enjoying the symposium.
God, it is.
Okay, we have a third speaker in this first session, and then we have a 30-minute break, and then we'll come back for the second session in our Q&A panel. Our next speaker is
Could we ask
Dr.
the second person to please stop sharing their screen?
Sure.
Oh, yes, Brendan, can you please stop sharing your screen so that Joanne can share hers?
Thank you,
Thank you.
Brandon.
All right, our next speaker is Joanne Woiak. Joanne is Dr. Joanne Woiak. Joanne is an associate teaching professor in the Department of Disability Studies in the College of Arts and Sciences at the University of Washington in Seattle, which is also my alma mater for my PhD. Her research interests encompass the social justice implications of knowledge about genetics, reproduction, and health in the modern era. Her focus has been on the history and perception of disability in American and British history and culture, including the history of sterilization and constructions of mental disability. She's been very active in developing symposia and resources on the topics of eugenics and disabilities. So Joanne, please take it away.
Thank you, Kelly. This is Joanne Woiak. Thank you, Kelly and the SSE for inviting me today. And thank you to Brandon and Brian for, wow, what incredibly powerful talks to follow here. So I'm going to focus on history of eugenics and through a disability studies lens. And I'm really happy to have this opportunity to share this perspective with you all, with this audience here. I do teach at the University of Washington in the Disability Studies program. So the question that my field, disability studies, asks is, what is disability, right? We are sort of intervening across the board, across the curriculum, to look at the ways that typically disability has been constructed as a biological problem, as a medical problem, and to say, hey, actually, if we look more broadly, how is it that society constructs disability, right? So I'll show and talk a little bit about that framing. As far as myself coming into chatting with you all today and into my field and my teaching and my research in disability studies and where it intersects with history of eugenics, and I'm just to say, I'm gonna, for my 30 minutes, mostly unpack with you about what is disability studies and why is it, and how should it inform our understanding of eugenics. So I'm not gonna unpack a lot of the details of eugenics because I think that is familiar to you all. But I'm coming into this as a person who got their PhD in the history of science and medicine, and had been looking at the history of the British eugenics movement, a particular corner of it, a debate that Carl Pearson was having about whether alcoholism impacted offspring or not, and what that meant for, quote unquote, the future of the race. And when I came over here to Seattle, to University of Washington, and started teaching a history of eugenics class and teaching some history of science, I met up with the community that became my home, became my love, which is the disability community here at University of Washington. And I identify as a non-disabled person, so I always want to put that out there. I think it's something that needs to be troubled in my field of disability studies, that academia is incredibly ableist, and most of us who are doing the research and teaching identify as abled or non-disabled. We're not disabled folks. But I have, as many people do, family connections with disability. So I met this community and became a disability studies teacher and disability studies scholar, never even realizing that I was deploying a critical lens on disability all along, but without naming it. And I think that's a big part of what the history of the eugenics movement has been. and sort of that, or the history of sort of doing the history of eugenics. The history of doing the history of eugenics is to not actually recognize that disability is a category of oppression, is a category of analysis, is an identity, and to sort of gloss over that part. So that's why I've been highlighting it here. And I also made a note for myself here to say that when I came into disability studies, and still having been teaching it for 15 something years, I am learning with and from my students. Again, I'm not at that central position of the field as a non-disabled person, so I'm always learning with the students. So speaking of the students, yesterday was the last day of class over here at University of Washington, and I taught my history of eugenics class this quarter with 50 students, which was a lot. And it is a combination of folks from the sort of social sciences, humanities side, a lot of them in my field of disability studies, and my class is cross-listed with the bioethics and humanities department in the School of Medicine. So I'm reaching a lot of pre-med students, health science students, public health students. And what I'm realizing is that I'm sort of sneaking some critical disability studies into the curriculum of those students in the health sciences in the guise of this history of eugenics class. They often don't know what they're getting into because it's just called history of eugenics. And they're not getting disability in other areas of their curriculum. So I was chatting with a student after class, as they tend to stay on the last day and tell you what they thought about it, and for better or for worse, right? And what they got out of it. And my student said to me, you know, she had been thinking about how impactful this was to sort of uncover her own ableism, right? Her own assumptions about disability, which had never been sort of centered before in her thinking. So she was thinking about why isn't disability in her other public health classes, other health science classes, or elsewhere in the curriculum. And then she came out of this really thinking differently about what is eugenics and what is the history of eugenics. Why does this history matter so much? She was saying, I had never thought about prenatal testing from a critical perspective, that the disability community is critical of that. And so she thought differently. We all were thinking differently about how marginalized and multiply marginalized communities may understand what eugenics is. And I'm thinking that too, right? It's more complicated than sort of a straightforward story of the histories of biological determinism and of restricting people's reproductive rights. It's a big part of it. But for disability community, there is a kind of a bigger picture here about the diminishing of the value of life, right? The assumption that certain lives are too much of a burden and not valuable. And that is really a bigger definition of eugenics that I think is really, really important right now. So just a very, very briefly version of disability studies. Disability studies as a field is reframing and reclaiming what is disability. And this is a graphic easily found on the web, which sets up the sort of dichotomy here in the frameworks. How do we think about how disability is constructed in society? On the left is the medical model of disability. Sometimes I call that the individual model, that says the individual person's body is the problem, the impairment is the problem, and that impairment, that difference should be fixed. And that is contrasted with what's on the right side and through the graphic where you see that the stairs are the problem, right? For, in that case, a wheelchair user, it is the built environment, it is social attitudes, it is policies that do not include disabled people, that do not think about differences in bodies and minds, that don't value those differences. It's society, it's the environment that's the problem. And having said that, I'm really reflecting deeply here on Brandon's points about the appreciation for genetics. And I want to reiterate that here in disability studies, that we have to have also an appreciation for medicine at the same time that we're critiquing the pathologization and the medicalization of disability, right? So as long as there is, disability studies is arguing for space for power sharing between disabled people and medical practitioners. And certainly there are disabled people who are seeking cures and treatments and so on, right? But it is the lack of power, is the lack of choice within those systems that is being critiqued when we think about the medical model of disability. So I think of disability studies almost could call itself normalcy studies, right? And it's very adjacent to, say, queer theory and queer studies, where what we're doing is calling into question the very idea of normalcy and thinking about how normal is enforced and how it is oppressive for so many of us. So as disability studies scholar Leonard Davis puts it, quote, "The problem is not the disabled person," right? It's not the individual or medical model. The problem is the way that normalcy is constructed to create the problem of the disabled person. So we have to always be thinking about, where does normal come from and why do we live under these regimes? And just very quickly, I'm going to mostly use disability first language here, which is identity and pride first language. That's very common in disability studies. It's very social model based, right? To say that it is society that disables me, so I'm a disabled person. But many of us in this group are also familiar with, and it's very important to use person first language in many settings, people with disabilities. And I also want to name the other category, which is non-disabled or abled, right? I think that that centers disability. It's really important to do that in DS conversations, and it's sort of like marking whiteness. As DS scholar Stumi Linton says, these designations, as reclaimed by the community, are used to identify us as a constituency, to serve our needs for unity and identity, and function as a place for political activism. So let's talk about eugenics, obviously. And I'm thinking about why disability studies is important for historians of eugenics and for all of us who want to critically reflect on that history. And as my colleagues here are saying, what are the calls to action, against the current versions of eugenics? And I think there's a history of disabled people related to the eugenics movement, which maybe is the more obvious side of this, maybe not. And I'll just say a little bit about some of the work and some of the directions on disabled people impacted by eugenics historically. And I haven't said, but I'm really here talking about the history of the United States eugenics movement. It's going to be familiar, maybe more familiar to many folks, the history of Germany, the history of the Nazi genocides against Jewish people and disabled people. And I'm talking here mostly about the US with my examples. So there's people with disabilities. Then there is the idea of disability, right? The concept of disability. And once you center that category of analysis and oppression, how does that open up some new ways of thinking about the past, our eugenic past, and the consequences today of that past? So the history of people with disabilities, just to give one example here, I have on the right is a map that circulated in the 1930s, and you see this a lot when folks give history of eugenics lectures. The states that have hashtags or black marks are ones that had, as of the 1930s, forced sterilization laws, right? Compulsory sterilization, which said that people in certain types of institutions for people with disabilities could have surgery performed on them, regardless of their choices about whether they wanted to continue to have children or not. And we know that more than 30 states had these laws. I've got the verbiage here of Washington State's 1921 sterilization law, which said the superintendents of all state mental hospitals and custodial schools must report all feeble-minded, insane, epileptic, habitual criminals, moral degenerates, and sexual perverts who are persons potential to producing offspring who, because of inheritance of inferior or antisocial traits, would probably become a social menace or wards of the state. So pretty good synopsis right there of what the eugenics movement was trying to do. The idea here of disability, of being a burden, of being incapable of contributing to society, and disability sort of underlies these other categories, right? About sexual difference, sexual deviance, so-called criminality. And some of the work that Kelly alluded to, we did a big symposium in 2009, sorry, here at University of Washington, because 1909 was Washington State's first eugenics law. Oops, we got, Are we okay there?
We can still hear you.
Okay,
And
just
see
double checking.
the
No, I
slides.
saw a hand. I thought maybe I needed to pause. Sorry about that. All. Okay, sorry, thank you. We had Washington State was the second state in the country that passed a eugenic sterilization law before California even. And as you probably know, California did most of the sterilizations. Sorry about that, Joanna. No worries. Seeing the chat. Yeah, so we had a sterilization law on the books here, and I actually just discovered that it looks like our legislature is overturning that 1909 law as of this summer. It was still on the books, right? To sterilize people who were defined as quote unquote habitual criminals or quote unquote sexual perverts. But we had the second sterilization law that came in that was this one, the verbiage I have was more typical of the sort of scope of who was having this violation of their rights during the eugenics era, the early part of the century, in that it was for people who were specifically incarcerated in institutions for people who were defined as having intellectual or mental disabilities, or people defined as having mental illness at this time. And I've done a little bit of going to the state archives here and looking around at what records are existing. And as some of you may know that Alexandra Stern also has the Sterilization and Social Justice Lab, right, where they have been sort of crunching the data on a lot of these records of who got sterilized across different states. So it's work that I have not actually published, but it's this history of who was defined as disabled, what were the reasons that they were sterilized, and so on. What were the reasons that they were incarcerated? Because I think that remembering that the history of eugenics is also a history of confining, segregating, removal of people with disabilities from the community, from society. And it's a history of violating their reproductive rights. And both of those pieces are important. Also thinking about the history of disabled people and why centering their histories is important in eugenics history is to think about how is normal socially constructed. So, going back into the 19th century, thinking about industrial capitalism, the rise of universal education, which are powerful systems, but that also sort of othered groups of people, right? Defined who was now unproductive. And disability becomes identified as a category, including as a sort of policy category here in the 19th century, who is a burden, who is not capable of working, right? And then the history of IQ testing starting in the very earliest part of the 20th century. By the 1910s, United States eugenicists had sort of taken over this history and said that intelligence is not something that you can develop, but instead it is hereditary, it's unchangeable. And this category of the feeble-minded becomes created and sort of really solidified here in the early 20th century as defined as a group of people around disability who now are a menace to society and are sort of, again, at the foundation of thinking about the reasons that we have these other quote unquote social problems that here, biologist Charles Davenport, founder of the Eugenics Record Office, says it's a reproach to our intelligence that we as a people should have to support about 500,000 insane, feeble-minded, epileptic, blind and deaf, 80,000 prisoners, 100,000 paupers at the cost, right? Again, this is the very heart of eugenics, this economic argument of $100 million per year. I'm thinking more broadly about why disability matters here and why a disability studies lens that takes a social model perspective on disability matters. There's a really impactful article recommended to everyone, happy to share it, by a disability historian named Douglas Bainton from 2001, Disability and the Justification of Inequality in American History. So how was disability as an idea, as a concept, not necessarily as a lived experience, used to discriminate, to exclude lots of people, lots of groups of people in the late 19th and early 20th century? So as Bainton said, not only has it been considered justifiable to treat disabled people unequally, but the concept of disability has been used to justify discrimination against other groups by attributing disability to them. And the three examples that Bainton highlights are citizenship debates over gender, over universal, or over suffrage, women's suffrage, about race coming out of the Civil War, and then about immigration and ethnicity. And it's different groups, women, Black folks, immigrants from various parts of Europe in particular, were getting sort of defined, categorized as disabled in the sense, in a medicalized sense of deficient, as defective, right? That sort of medical model meaning of disability was getting applied in order to deny these growing demands for people's rights and justice, right? Whereas citizens, proper citizens, non-disabled, white, middle-class men were marked as able, independent, rational, and productive. So when disability gets used against other groups, sort of weaponized, so to speak, there is I want to highlight here also, or at least bring into conversation that my field of disability studies really needs to, and is in parts, we are engaging more with the perspectives, the knowledge, the wisdom of multiply marginalized disabled people. In particular, Black, Indigenous, people of color who are disabled, queer, gender non-conforming, people who are disabled. And T.L. Lewis, who's a Black disabled lawyer, disability justice activist, has put forward a definition of ableism through a disability justice lens, which really gets more to this intersectional meanings of disability, right? And the leadership of folks who are most impacted by these multiple systems of oppression. So I think that disability studies is incorporating more of this disability justice view. That I am, you know, I want to share it with my students. I want to share it with you all, but also always recognizing that I am in this very, you know, as a white non-disabled person in a position of great power, as an academic, and not to be sort of appropriating this knowledge, but the knowledge of multiply marginalized disabled people really is pushing forward what disability studies is. And I think it's pushing forward how we're thinking about what eugenics was and what eugenics is. So all of that is to say, look up T.L. Lewis, T.L.'s work on ableism. T.L. says a system of ableism is a system of assigning value to people's bodies and minds based on societally constructed ideas of normalcy, productivity, desirability, intelligence, excellence, and fitness. And then there's much more details here about white supremacy and other forms of oppression and violence. And then TL says, you do not have to be disabled to experience ableism. And I really think that that, you know, dovetails with the disability studies, Douglas Bainton analysis from earlier, and thinking about why it's so important to put the history of eugenics into this broader context of forms of exclusion, violence, oppression against many folks in society. So some of the ways historically that this sort of intersectional thinking about how disability intertwined with white supremacy, intertwined with patriarchy, intertwined with classism, is to think a few examples here. What's the role of, what was one of the roles of gender in the history of eugenics? It's notable that more than 50%, as much as 60% of the recorded 60,000 Americans who were forcibly sterilized. And I do say recorded because that's just what the Human Betterment Foundation collected from every state, from every institution. If you go to the state archives, say in Washington, you will find out that there were hundreds of women during the early part of the 20th century who were having hysterectomies done, right? So there were eugenic practices happening outside of the confines of the law as well. But mostly it was girls and women who were being sterilized. Think about the case of Carrie Buck, right, which is probably familiar to many of us. The Supreme Court ruled 3 generations of imbeciles are enough, that it was legitimate to take away a person's right to privacy, right to their own choices about their reproductive lives, and have them sterilized in Buck versus Bell. And the ways that Carrie Buck lived at these precarious intersections of systems of oppression, that she was poor, she's from the South, She was a teenager at the time that she experienced sexual violence, which resulted in her having a child. And from her perceived sexual deviance, because of the violence that happened to her, right? She got labeled as disabled. She got labeled as the so-called feeble-minded. And the Supreme Court said, let's compare this to compulsory vaccination. And it is for the good of the health of the state, right? It's for the good of people in general, like a public health measure that we should be sterilizing. Who are the people, right? Whose good is this and who is being left behind? These are the questions that we continue to ask about where are the implications of eugenics today? Where does it live on? Thinking about ethnicity and the history of immigration, this is an image from probably about 1918 of an immigration officer, and then a translator on the left, and on the right is a person at Ellis Island trying to come into the United States to move, and he's being given one of these intelligence tests. They're testing people from Southern Europeans, Eastern European origins, Jewish folks, and so on, right? And they're finding that as many as 80% of folks from these national origins are measuring as feeble-minded or morons, according to these scales, and according to these incredibly racist, culturally biased, class-biased tests that are being given. Groups are being ranked, right? Just as what we were talking about in the prior talks, that it's assumed that there are genetic differences between ethnic groups, and those assumed differences then are getting used, and the concept of disability, that these folks have mental disability, then is used to shore up and justify very explicitly the very strict immigration restrictions that came in 1924, which had within a decade had the impact of preventing many Jewish people from immigrating into the United States, and obviously exacerbating the genocide at that point of the 1940s. And just thinking, I mean, there's a lot obviously to be said about the intersectionality, the intertwining, the co-construction of racism and ableism in this history. But just to come back a little bit to this history of sterilization, I mentioned about the Buck versus Bell case, and there is a history of poor white victims, and that many of the victims were female, but also thinking about there are histories that are less told of the forced sterilization of Black Americans. In North Carolina, for instance, there is initially, it was largely white folks who were being sterilized, but by the 1960s, because these laws and these practices did continue, as much as 64% of the victims were African American. Many of them were welfare recipients. North Carolina's history is also interesting because a lot of the folks who were sterilized were not actually incarcerated in the disability institutions. They were coming through the welfare system. And it's an interesting history, which I also see in Washington State, of the complexities of some folks who actually tried to utilize the apparatus of the sterilization laws in order to get birth control for themselves, right? It's a very, very complicated history when you didn't have other choices. But having said that, by and large, the white supremacy of North Carolina's program needs to be more researched and understood. Similarly, in California, we know that that is the state that sterilized the largest number of people, as many as 20,000 up to the 1970s, and Mexican origin folks and African Americans were operated on rates that far exceeded proportions in the general population. And perhaps famously in the 1970s, LA County Hospital coercively sterilized hundreds of Latinas, and 10 of them filed a lawsuit, which unfortunately did not succeed, but we got federal regulations around sterilization put into place. And wrapping up, definitely, thank you. Again, the calls to action are out there, the hopes, the promise for pushing back against the modern forms of eugenics. There is I've noticed in the last 2-3 years a real flourishing of an anti-eugenics website, of websites, of conferences. I can easily pull up and share lots of examples with you. Just another example to give is right here in Washington State. A sister university here, University of Puget Sound, just got the university to remove the name of a eugenicist from the museum, right? It was named after Slater, and they removed it, and we had done a symposium about that. So there's a strong anti-eugenics movement happening. This is an example of Indigenous folks who are doing this work, The Witness Tree by Judy Dow, artist and educator of Abenaki and French Canadian descent. And it's a sort of a play on the famous eugenics tree with all of the disciplines that supposedly built up, the theory and the practice of eugenics. And instead, she points to love, memory, trust, found voices, justice, respect, education, support, and more as what we need to do for anti-eugenics. The pandemic and eugenics, much to say about that. But I think, again, here, this is where disability justice folks thinking about the ways that disability, that during the pandemic, we need to think about eugenics in a more expansive way as the devaluing of human life. And we saw that initially with literally the removal of ventilators from disabled folks. And we sort of see that in the continuing on with the removing of mask mandates and so on. And the disability community is very, very attentive to that. So thank you, Mike, for the timing. Thank you all for being here to talk about disability, also a little bit into this conversation. I look forward to seeing you all in the next session. Thank you.
Okay, welcome back, everyone. Good to see everybody back, and great that the interest in this symposium is so high. I have a very quick announcement first. Just a reminder that the SSE and the Tri-Societies have a code of conduct, and that applies to our virtual meeting as well as our in-person meeting. So if you see anything that needs reporting, we actually do have an evolution safety officer who is available online. So you can use of that resource if you need to. Our next speaker is Dr. Graham Coop. Graham is a professor in the Department of Evolution and Ecology at UC Davis. His lab research is focused on understanding the evolutionary forces that have shaped genetic diversity in various species, including humans. They do so by blending modeling and data analysis. Graham has interest in traits, their genomic basis, and he's written extensively lately on what genomics can tell us or not about these traits, including in humans. So please, Graham, share that information with us.
I'll unmute myself. Thanks to the organizers for inviting me to talk today. I've enjoyed and learned a lot from the other talks in this symposium. I'm looking forward to our conversation later during the question and answer session. So, as we know, evolution and genetics, and in particular human evolution and human genetics, have a popular place in the public imagination. Many of us got into the fields of evolution and genetics in part because we were excited about the big questions that evolution and genetics could teach us about who we are and where we come from. And that's, a bigger source of excitement for our students and for our appeal to the public. The flip side of that is, as we know, our field has a strong legacy of reinforcing genetics and race essentialism, where scientists either accidentally or purposefully have introduced ideas from genetics into political debates in order to reinforce pre-existing hierarchies. I'd argue that as a community, geneticists and evolutionary biologists, for our excitement about our field, can still fall into the trap of overplaying the role of genetics, genetic differences, and adaptation in human variation. So that's the issue that we face. I'd argue also that there's an exciting opportunity for us to change the ways in which we talk about our field, both as scientists to each other and in discussions with the public. But on a more basic level, there's an opportunity for us to revisit the research questions and the research frameworks in which we tackle problems. So I'm a human population geneticist, I'm a human evolutionary geneticist, and through my research, I'm broadly interested in the ways that we teach, think about, and discuss genetic variation. And in today's talk, I want to touch on two ways in which we're moving things forward in a small way, in terms of thinking about how we visualize human genetic variation and in the use of human ancestry groups as sample descriptors. So taking a step back, in population genetics and evolutionary genetics, we often make use of simple models where populations persist through time in particular geographical locations. Those models are incredibly useful to us to inform our intuition. However, those discrete populations don't exist out in the real world. They're a modeling construct, and it's too easy for us to think about populations in an overly discrete way. The reality is that we're all related to each other very closely, by varying extents, for an unimaginably complicated genealogical tree. As a consequence of that, patterns of genealogical relatedness and genetic similarity vary fairly smoothly across people, shaped in interesting ways by geography and population history, often in ways that are correlated with environments, complicating the task of human genetics greatly in separating genetic and environmental interactions. As a consequence of the fact that patterns of variation vary at all different scales, there's no single right level for us to describe population genetic structure in populations such as humans. There's variation in population structure, both within families, among geographical regions within countries, among continents, across the world. The level at which we describe genetic structure will depend upon the framework and the problem that we're addressing. And as a consequence of that, all verbal descriptions of population genetic structure are necessarily incomplete and open to misinterpretation. So sort of broad backgrounds. The difficulty is then, when we're talking about genetic variation in the classroom, we need tools to put across ideas about human genetic variation accurately in ways that students can quickly grapple with while dealing with a range of complicated material. Often when we talk about population genetic structure, the ways that we visualize population genetic structure can overemphasize differences amongst individuals and obscure the broad genetic homogeneity that exists amongst human people. So in order to try and address that, James Kitchen in my lab and I have been working on ways of visualizing human genetic variation that are appropriate for various levels of use in the classroom. And in doing that, we're building on some really nice work by Donovan and others, and a framework put forward by John in John Novembre's lab, and building on pre-existing ideas about how to better display genetic variation. The resource that we're going to do to use this is we're going to be making use of an often used human genetic data set, the 1000 genome sample, which is a broad sampling of people to provide a reference data set for human genetic variation. We're going to be zooming in and making use of the Americas samples in the 1000 genomes data sets. So our genomes are big places. There are over 3 billion base pairs in our genomes. And when you go to a resource like the 1000 genomes data sets, around 2.9 billion bases of the genome are accessible to the sequencing technologies used by the 1000 genomes. We can then think about the 1000 genomes Americas data sets and think about the variation that's assayed by those technologies. And what we see is that if we take all of the observed base pairs that have been seen to vary in the 1000 genomes Americas samples, some 600 odd genomes, there's around 40 million sites where variation occurs against that much larger background of 3 billion sites. So we already see at this scale that humans are very genetically similar to each other. There's relatively low levels of polymorphism, even when you go out and sequence hundreds of genomes. Okay. The thing is that many of those sites are rare variants where one individual may carry a T and many other individuals, maybe the entire sample, carry an A. So most of the variation that's present in human populations is rare, That's interesting for lots of different reasons, but those aren't common differences amongst individuals. And so what we can do is we can focus in on common differences amongst individuals using an arbitrary cutoff of 5% as the minor allele frequency. So we can look at sites where an allele is present at more than 5% frequency in the population. And what we see there is that there's some 10 million sites where there's a variant above 5% frequency that we take as a common variance. So we're seeing, against this huge human genome of some 3 billion base pairs, there's a relatively small fraction of sites where there are common differences amongst individuals within this sample from the Americas. What we can do is we can break out our samples by their different sample labels. In going out and collecting 1,000 genomes, they were collected from particular geographic regions, from particular populations, and we can look at the variation present in this. So we have samples, for example, from Puerto Rico, samples of people of European ancestry from Utah. We have people from African Caribbean people from Barbados. And what we're doing here is the size of the circle depicts the number of common variants present in those populations, these variants above 5% frequency. So what we can see is that human populations differ somewhat, human samples differ somewhat in their levels of common variation. Populations, samples whose ancestors lived outside Africa historically have lower levels of common genetic variation than populations or population samples where their ancestors are drawn from Africa and have experienced a larger effective population size through time. So we can then ask, well, how is this variation shared amongst populations? And to do that, we can look at the overlap in common variation amongst these population samples. Here you're looking at a Venn diagram, or more technically an Euler diagram, where the area of overlap indicates the proportion of variants that are shared between different samples. You can see this strong overlap in variation amongst all of these samples from the Americas, emphasizing the fact that most of this common variation is shared amongst these populations. We can also see interesting aspects of population history already starting to emerge from this approach. So for example, those samples which have experienced historically, their ancestors experienced historically, population bottlenecks, such as those individuals whose ancestors lived in Europe, have lower levels of genetic variation. We see those samples whose ancestors lived in Western Africa, for example, those individuals drawn from the Afro-Caribbean sample, those individuals have a larger set of genetic diversity. It overlaps nearly all of the genetic diversity present in the other samples, but there are variants which are common in those populations and rare or absent in these other samples. So we get this nice depiction both of the sharing of this common variation amongst these samples and also differences among samples in the sharing patterns that we see. We can zoom out and look at samples from across the 1,000 genomes regions, looking at the samples which have been collected from Africa, from Europe, from South Asia, from East Asia, and the Americas. And we can see that the samples drawn from Africa have this characteristic higher level of genetic diversity. And when we look at samples taken whose ancestors lived in Europe, there's a lower level of genetic diversity within those populations, but there's this very strong level of overlap within each one of these geographic regions, such that when we look at our samples within the Americas, we're seeing that overlap nicely shown. What we can do then is we see this strong overlap in genetic variation amongst these samples taken from the Americas, and we can place it back in the context of the entire human genome. That human genome of 3 billion base pairs, we can see that that overlap in genetic variation, common genetic variation, is against a much broader background of the homogeneity of the human genome amongst individuals. As human geneticists, as evolutionary biologists, we're fascinated by genetic variation and in telling stories about that. But it's important for our students to keep in mind that those stories about genetic variation are against a much broader background of genetic homogeneity, a high level of genetic homogeneity amongst people. In the classroom, we can then go on, building on that background, to talk about how we lose genetic variation, to talk about human evolutionary history, to talk about local adaptation, to talk about the genetic and environmental contributions to differences in disease risk and phenotypic outcomes, but against that background of a broad sharing of the genetic variation of humanity. Okay, so what I'd like to do in the second part of my talk is move on to talk about other ways in which we talk about genetic variation and talk about these common descriptors of genetic variation, genetic ancestry groups. So human geneticists often talk about genetic ancestry groups, talk, for example, about European ancestry, talk about East Asian ancestry, talk about African ancestry. And those are labels that people use to describe genetic variation. And indeed, myself, I use those variables and those labels. But I want to talk about how we can move to doing a better job about talking about genetic variation. And this echoes the calls of others to become more precise about how we talk about genetic ancestry and to emphasize the more sort of continuous nature of genetic ancestry across others. Recently, I've put forward this argument that actually as a field, as human genetics, should move away from talking about genetic ancestry groups to talking about genetic similarity labels as a sample descriptor. And a similar argument has been put forward by a National Academy committee discussing the use of population descriptors in genetics and genomics research. And so this is a really useful time for us to reflect about the labels that we use to describe human genetic variation. It's necessary for us to use verbal descriptions of genetic variation, but what are better ways of us to engage in doing that? And in doing this, we're talking about human genetic variation, but I want to emphasize that many of the approaches that we use to describe human genetic ancestry have wide application in non-human applications in evolutionary biology. And so the conversations that we're having in human genetics about how to talk about genetic variation also have important echoes in non-human applications. Okay, so in doing that, I want to emphasize a strong distinction between two related concepts. The first is in that there's a precise definition of talking about genetic ancestors versus this much squishier matter of talking about genetic ancestry groups. When we think about our genetic ancestors, that's the subset of individuals who you've inherited your genome from. So for example, around 450 years ago, your family tree has doubled each generation we move back, and you have possibly have over 32,000 living genealogical ancestors 450 years ago. But you've inherited genetic material from only a small fraction of your ancestors. So the first thing to note is there's an important distinction as we move back into the past between your genealogical ancestors and your genetic ancestors. Your genetic ancestors are just a small subset of your genealogical ancestors, and that subset gets dwarfed as we move further and further in past, as your genealogical ancestors grows much more rapidly than your genetic ancestors. When we're looking at population genomic data, that population genomic data informs us about patterns of relatedness. And as a consequence of that, we're learning about our shared genetic ancestors and our relationships to each other through those shared genetic ancestors. We have sophisticated methods to describe some of the properties of our genetic ancestors, thanks to advances in statistical population genomics. A really wonderful set of techniques, including constructing genealogical trees or whole ancestral recombination graphs to describe our patterns of genetic relationships amongst individuals. So we have these sophisticated statistical tools, but our interpretation of the patterns that we see in genetic relatedness in terms of talking about our genetic ancestors comes from the geographical and sample descriptors that have been associated with a particular set of samples that we're analyzing. The investigators who collected those sample descriptors and geographical labels have particular priorities in collecting those labels. And so the interpretations that we place on the data that we examine are necessarily partially a social construct based on the sample descriptors that we are receiving as part of analyzing particular data sets. So our methods get ever more sophisticated, but they're closely tied in their interpretation to this social construct of the labels that people attach to samples. One common sample descriptor that people make use of is that of genetic ancestry group. So people talk about European genetic ancestry, East Asian genetic ancestry, African genetic ancestry. What do we mean by that? Well, what we mean by that, or what people mean by that, is that that's nearly always based on a measure of genetic similarity, where we're using some measure of genetic similarities to see who you're genetically similar to, and then borrowing labels across individuals in order to label individuals by a particular type, for example, European genetic ancestry. So to see that, let's think about using, for example, principal components, one common way to think about genetic ancestry. These principal components are these major axes of genetic variation. Here you're looking at the plot of principal component 1 versus principal component 2, and each dot here is a sample member of the 1,000 genomes with their location on principal component 1 and principal component 2 plotted against each other. And these individuals are colored by the reference panel that they're drawn from in the 1,000 genomes. Note that these population labels explain relatively a small percentage of the genetic variants present in these samples, again emphasizing the fact that humans are very genetically similar, and these major axes of variation, while interesting, are only a small part of the story of our genetic variation. Okay, so if you projected my genotype onto this graph of the principal components of the 1,000 genomes, I'd almost surely fall close to these other individuals labeled as coming from the European subsets of the 1,000 genomes. And on the basis of that, a human genetics investigator would probably label me as having European genetic ancestry. Now, that might be a perfectly reasonable way of labeling me, but it's a construct based on the labels that are present in the 1,000 genomes. The statement really is that I am genetically similar on these first two principal components to other individuals who've been labeled in the 1,000 Genomes Project as belonging to the 1,000 Genomes European population subsets. Okay, so these labels of genetic ancestry are nearly always based on measures of genetic similarity. For example, who a person clusters with on a principal component plot. The thing to realize is that when we look at large data sets of people collected from large communities, we see huge variation in where people fall on principal components plots or other measures of genetic similarity. So here you're looking at a data set taken from Lewis et al. Of 31,000 individuals projected onto the principal components. Those are the gray individuals here, projected against the 1,000 genome samples, again on a similar plot of PC1 versus PC2. Here you see this continuous nature of genetic variation, where there's a broad spread of individuals on these measures of genetic similarity, again speaking to the difficulty in constructing discrete categories for human genetic variation. There's continuous variation in measures of genetic similarity here. Okay, there's a broad set of other approaches that population geneticists have developed to talk about genetic ancestry. So just beyond where an individual falls on a principal component plot, we also see clustering approaches that are structure-like, where an individual's ancestry is broken down in terms of the proportion of their genome that comes from some hypothetical sets of discrete clusters. For example, an individual may be described as having 60% African ancestry, 40% European ancestry, but really what that's saying is that 40% of this individual's genome is genetically clustering, is genetically similar to another sample or a reference sample that's been labeled as having European, as being European. We also see more sophisticated approaches that will paint an individual's genome as being drawn from a set of reference ancestries. For example, this individual, Caribbean descent individual, is having their genome as being painted as being drawn from European ancestry, Native American ancestry, or sub-Saharan African ancestry. Again, these statements of ancestry are really a statement of genetic similarity, of similarity to some reference sets of samples that we've chosen to label in a particular way. So all of these statements about genetic similarity, about genetic ancestry, are really statements about genetic similarity. The hypotheses they put forward, if specified in terms of geographical locations of where an individual's genetic ancestors may have lived, may be reasonable hypotheses, but they're based on relatively simple models. And the question is can we separate our ideas about labels from those ideas about genetic similarity? So there are many common issues that have been raised in these discussions of genetic ancestry labels. A very common and fair criticism of these approaches to genetic ancestry is that the ancestry labels that we use are often based on very broad continental labels, such as African ancestry, European ancestry, East Asian ancestry, and overlap strongly with racial categories, which creates a lot of confusion and between those ideas of ancestry and race. On a more technical side, the resolution and identities of the ancestries that we label individuals with are a function of the reference samples that we choose to include. And so we're constructing ideas about ancestry through the population samples that we choose to include in our analyses. And as a consequence, these labels are always somewhat arbitrary. The statements that we make about ancestry are also usually bracketed to a specific time, usually a few 600 years ago to a few thousand years ago. However, that time bracket that we're specifying is often left unsaid, and it's often unclear why we've settled on that particular bracket of time in order to talk about ancestry. The ancestries that we're constructing are at best statistical modeling constructs, but quickly can become rarefied in the public's mind, and indeed scientists' mind, that can often obscure important questions from view. So these ideas about genetic ancestry can accidentally obscure inhomogeneity within ancestries and also obscure the continuum of relatedness across ancestries. In light of that, a number of different responses have been put forward, all of which are valid. One call is to move towards finer grain scale ancestry, so to move away from these continental labels that are so problematic and move towards more fine geographical scales of relatedness. So if you look at my 23andMe profile last year, you would see that my ancestry has been broken down into European, into Northwestern European, into British and Irish, into Greater London, and all sorts of sub-regions within the UK. However, if we're going to have issues talking about ancestry at the scale of entire continents, we're even less sure about what we're talking about when we talk about Greater London ancestry. And so it's difficult for us to be precise about what we mean. We can also think about slicing ancestry into different time periods in the past. We can have ideas, and we've worked on this ourselves, of tracing genetic ancestors back across geographical space. Now, all of these more complex ideas are again statements about genetic similarity. Phrased in terms of genetic ancestors, many of these ideas may be scientifically useful to human evolutionary geneticists, population geneticists, and genetic anthropologists interested in human evolution. However, the vast majority of research in human genetics isn't focused on these basic questions of population history. Rather, I'd argue that most of human genetics cares more about correctly controlling for genetic similarity in testing for hypotheses about genetic variation. Human genetics is concerned with correctly matching cases and controls, for example, and they're doing that on the basis of genetic similarity, not some mushy idea about who an individual's genetic ancestors were. So on the basis of that, I and others are arguing that we should move away from these ideas of genetic ancestry groups and move towards just simple ideas about genetic similarity. And those ideas have been much more developed in this National Academy Committee report on population descriptors. The argument is that we should move towards simple statements about genetic similarity to say, for example, that I am genetically similar to individuals from the 1,000 Genomes Great Britain sample, rather than describing me as having European ancestry. On the basis of that, I can also be specific about the method I use. I can say that I'm comparing myself to the 1,000 Genomes project, for example, using principal components, and so be far more precise about how I'm assigning a label that I'm genetically similar to some group. Now, that's obviously a mouthful, but we can move towards more simplified language and, for example, say that I am like the 1,000 Genomes Great British sample, accompanied in the text, along with a clear definition of what we mean when we talk about that. Importantly, beyond just improving the precision of our language, I think it also enforces precision on us in terms of the scientific questions we ask. In moving towards measures of genetic similarity and describing human genetic variation, the question becomes how similar a match we are making when we're talking about genetic similarity, and by what measure. So we're forced to actually engage with what degree of precision of scale are we making when we're making calls about how we label individuals. It puts a focus on the panels that we are using to judge similarity. Importantly, that doesn't homogenize when we use these similarity labels. It doesn't homogenize as much within labels. To say an individual is the same as is not to say that they are genetically identical to. And so that's a useful set. For us as evolutionary biologists, moving towards this framework is important because it allows us to separate the problem of making genealogical, historical, and evolutionary inferences from the task of how human geneticists describe samples at a basic level. So my take-home message is here that there are important opportunities for us to accurately convey human genetic variation in the classroom and in public discussions, that one part of that is to move away from the use of genetic ancestry descriptors. And more broadly, we all need to continue our push to accurately represent human genetic variation and discuss the frameworks, the assumptions, and the questions that motivate our fields. And with that, I'm happy to end my talk. Thanks, everyone.
Thank you, Graham. Some common themes showing up here. Just to remind everybody, we have a question and answer panel coming up after our next speaker. So it's the third period in this session. And so we're asking everybody to hold questions until we get there. Our next speaker is Dr. Jed Carlson. Jed is a bioinformatician. He's interested in human evolutionary genetics, population genetics. He does bioinformatics for large-scale sequencing data. In addition to his human popgen research, he's particularly interested in the social impacts of his science and how it still is, to this day, misconstrued for use in inequitable social movements. He is currently a research associate here at UT Austin, which is also my institution. So I look forward to further discussions, Jed, when you're here. Please tell us about your research in this arena and your view on the way forward.
All right, thank you so much, and thank you for the invitation to speak, and yeah, engage with all of you on a topic near and dear to my heart. So, yeah, a little bit more about my background. So this really started, this interest started as a graduate student at University of Michigan, really just kind of spouting off a mouth on Twitter about perceived problems in how research was being consumed by far-right communities. And so this got some nice coverage from some media outlets, which really motivated me to kind of incorporate this more deeply into my own research. To Brandon's point about haunted houses, I also have some interesting personal anecdotes about kind of family history intersecting with the Aryan nations in northern Idaho, where my mom's family hails from. So these are pictures from neo-Nazi rally in Coeur d'Alene, Idaho in 1999. My family was actually, I grew up in Iowa, but my family was traveling there to see family, and we were there on that day. I remember my parents were, downtown when this was happening, and came back and were kind of concerned as you are. And I think that's kind of stuck with me as I'm really interested in kind of understanding how these groups are proliferating, and as a scientist, how they're incorporating scientific research into their ideology. So this talk was originally kind of developed following the shooting in Buffalo last May. As many of you know, the Buffalo shooter released a manifesto citing many instances of many scientific research publications and figures that really, I think, confronted a lot of scientists about, our ethical responsibilities to, mitigate that and I think in some ways absolve, in some ways distance ourselves from, being seen as complicit in racial violence motivated by our work. I do want to say, I think a lot of the opinions that came out after this were really focused on kind of a bioethical framing. If misappropriation is happening, we have an ethical responsibility to do something as scientists about it, whatever that might be, whether it's developing more ethical policies in IRBs and editorial boards and so on and so forth, or just kind of improving the way that we're conducting our own science. And I fully endorse those ideas. I think we're due for reforms on those fronts. But as Brian said at the top of the first session, I think maybe we need to reframe this as not a matter of miscommunicated science, but as more of an infodemic, to use a buzzword that's popped up alongside the COVID-19 pandemic. So, I think in my own work, I think we certainly need to situate these issues in an appropriate historical context. I'll go into that a little bit today. But also thinking about this in this kind of epidemiological framework of thinking about modes of exposure and transmission, how information is passing through different communities and infecting people in a sense. And my meta research work, I hope, really helps inform and improve how we as scientists are kind of interacting with this infodemic. And ultimately, I think we do bear responsibility to improve how we're conducting and communicating our research and coupling that with other areas of hopefully activism. So a little bit about the history here. If you're familiar with kind of history of race science, you may have come across the name Arthur de Gobineau. This is a French aristocrat who is kind of seen as one of the grandfathers of scientific racism. He published his essay on the inequality of human races in the 1850s. Another character by the name of Josiah Nott, he was a medical doctor in the US who was a major proponent of phrenology. James Hunt was an anthropologist in the UK around the same time, who was also publishing similarly racist tracts in his own way. A character that you may not be familiar with is a guy named Henry Hotz. So Henry Hotz was a Swiss or Austrian-American living in Alabama in the 1860s. In the 1860s, of course, things were happening in the US. So first of all, he helped Josiah Nott actually translate de Gobineau's work into English, exposing that to a much broader audience. And as kind of a connection to kind of the European contingent, Hotz was recruited to lead a delegation for the Confederates to London to solicit support from the intellectual community there for the Confederate cause. He did this mainly through running this pro-Confederate tabloid called The Index. And in that, it was imbued with all these kind of ideas of scientific racism. I think Hotz really helped unify these different schools of scientific racism coming from different countries and synthesizing those in a way that I think really set the stage for how scientific racism emerged in the early 20th century. So, this is kind of the deep origins of weaponized science as I would define it. We're going to skip over the kind of early 20th century stuff on eugenics and Nazi race science that hopefully all of us are fairly well familiar with. And I want to jump to another kind of less appreciated history here. Following World War II, if you're again familiar with the history of sociobiology and kind of controversy around E.O. Wilson, a lot of this actually centered around in the late 70s, a far-right group called the National Front published tracts basically saying, sociobiology is the new frontier of science. It reinforces all of our beliefs. Folks like Richard Lewontin and his wonderful ilk called them to task on that in a series of letters to the editor of Nature. But other groups around the same time were also embracing science as a means of promoting their propaganda. I forgot to mention at the beginning of this talk, there's a lot of racist content kind of shown throughout verbatim. So just general content warning for that, so it doesn't catch you by surprise. But here's a figure from the National States Rights Party in 1972. This was kind of a prototypical Nazi-inspired movement following the war. Another group, the National Alliance, they published this article in their tabloid called Sociobiology, The Truth at Last, similarly embracing sociobiology as kind of the fundamental scientific theory that motivated their beliefs. If you're not familiar with National Alliance, Southern Poverty Law Center describes them as the most dangerous, best organized, explicitly genocidal neo-Nazi movement in the latter half of the 20th century. And so this is really the origins of, I think, the Buffalo shooter and other extremists who have really embraced contemporary science to promote their ideology. So I'm going to just dissect some of the content of that manifesto and hopefully argue that this is something that we as scientists need to deeply consider going forward. So in the first few pages of the manifesto, he includes many different references to scientific papers. There's lots of figures kind of decontextualized, put in this kind of quasi-scientific, this kind of designed to look like a scientific poster. There's lots of figures and tables and this corpus of information that he's using to argue his ideology. In particular, a study that was, I think, has been kind of brought into the spotlight by the Buffalo shooter was a 2018 genome-wide association study for educational attainment. And like you do in any massive GWAS, you find hundreds, if not thousands, of statistically significant variants associated with the trait of interest. And so the table presented here, it's a little dry looking, I guess. Doesn't seem immediately compelling just at a glance, but the table shows basically a set of SNPs that were associated with educational attainment in this population, and then it appears to have cross-referenced those with allele frequencies from the 1,000 Genomes Project. All that to argue basically, these SNPs associated with greater educational attainment are much more common in Europeans, because of course they are, because that was how the study was designed, to look for variants associated with educational attainment in Europeans. So it's this very kind of convoluted and kind of deliberately misleading secondary analysis of published data. This is, again, all derived from a supplementary figure. So since I spend a lot of time kind of sifting through dark corners of the internet, I started looking around where this actually originated. And I found a thread on 4chan. So this is a common hangout space for the far right online. And about 6 weeks after the EA3 study was published, there was a thread of somebody basically laying out that the strategy displayed in the previous figure. The study of educational attainment supposedly insinuates that there's genetic basis for racial differences in intelligence. And you start looking down the thread, another poster says, going through this, the data is pretty damning when you check the effect sizes, but they basically posted kind of the prototype of this meme on September 15th. And you start looking around other 4chan threads around the same time, and you can see this evolving into different forms the way any meme does. People kind of remix things, they change around titles, they add annotations, and so on and so forth. And in these threads, there's actually explicit calls to use this sort of information as propaganda. All told, I've cataloged over 5,100 instances of these images being posted on 4chan alone, which I don't want to compare apples and oranges and say those citations are more meaningful than literature citations of this paper. But I think this certainly does speak to this is a tangible negative impact of this particular study that I don't think any of the authors could have foreseen or did foresee. I think they maybe could have, but all that to say, there's many other examples of memeified science. So here's another PCA plot that appeared on some bizarre site that kind of overlaid anime characters on top of that. I do want to point out for the non-human researchers here, measures of, say, FST between different species are often invoked to argue that different human populations are different subspecies. More PCA plots, more structure plots. There's kind of a whole ecosystem of scientific research figures being decontextualized and turned into memes in this way. In a recent commentary that I published in Nature with some colleagues, we actually looked at the kind of trajectories of these over time. And what we see is, there's pretty steady growth of, two different example memes that we cataloged. But notably, these seem to spike up around, notable offline events. So the murder of George Floyd, the 2020 election, the insurrection in 2021. And we didn't want to, read into this too deeply because it wasn't a peer-reviewed study. But lo and behold, a recent paper that came out earlier this year also looked at how online hate speech kind of corresponds to offline events, and they found very similar patterns of hate speech about race or anti-Semitism or immigration or religion and whatnot being kind of spiking in frequency following these offline events. So just to reinforce, I think these scientific figures are really being deployed as propaganda on these online spaces. And so I want to switch gears a little bit and talk about some of my meta research work on actually dissecting how these communities are consuming and engaging with scientific research in ways that I think, again, that the scientific community really needs to confront and contend with. So this is from mainly from a paper published in 2020 with my postdoc advisor at UW, Kelly Harris. So our motivation for this was to look at basically how papers are discussst on social media. So here's another preprint looking for GWAS meta-analysis of intelligence. You may be familiar with the altmetric landing page where it gives this colorful donut quantifying a score based on how many times the paper's been tweeted or posted on Wikipedia or kind of non-traditional citations outside of the scholarly literature. For this particular paper, you can actually scroll through the list of tweets that have referenced it. And so here's one from a guy named Kevin McDonald. If you're not familiar with Kevin McDonald, he has also earned his own SPLC profile, describing him as the neo-Nazi movement's favorite academic. He's a virulent anti-Semite who has tried to kind of intellectualize that belief in various ways. And so, this is just kind of the raw observation. We can see science being consumed and promoted by members of this community, but how do you actually analyze that systematically? So what we did was we collected, a corpus of 1,800 of the most highly tweeted bioarchive preprints at the time. Notably, just before the pandemic kicked off, I'd actually love to look at how COVID-related preprints have maybe been consumed in similar ways. But from this, we looked at all of the users who tweeted about these preprints and also incorporated information from all of their followers. And so it's a pretty massive data set. We had hundreds of millions of data points ultimately that were being analyzed. And I won't go into the full details of how we did this, but basically developed a method for doing this kind of computational audience segmentation on the groups that are on the people that are engaging with a given paper and trying to infer characteristics of them based on this large data set of information. And so we represented this in a way that we hope it's kind of intuitive to other evolutionary and population biologists. So you can think of this as analogous to a structure plot. So each horizontal stack of bars here represents one individual who tweeted about a paper. The color of the bar corresponds to the audience segments that we inferred, and the length of the bar corresponds to the kind of fractional assignment to those topics for that individual. So what immediately pops out for this particular study, this is looking at sex differences in gene expression in the human fetal brain. We have three audience sectors that are sort of explicitly far right. Topic 5 in orange here is white nationalist, MAGA, conservative, Trump, et cetera, and so on for the yellow and brown audience sectors here. All said and done, across the 1,800 preprints that we looked at, 70 of them had more than one audience sector with the keywords white and nationalist in kind of the top keyword sets. And when those audience sectors were present, those audience sectors accounted for as much as a quarter of the entire audience, which is, I think, pretty damning to scientists. Where I think we see the best in ourselves and the public that we're communicating to, that everyone's engaging with science in good faith. But I really don't think that's the case for a lot of papers, particularly those concerning controversial topics. Of course, not every individual is going to self-identify as a white nationalist on Twitter. And so we developed another method to kind of quantify this orthogonally. So we leaned on the kind of sociological concept of network homophily. So essentially, like follows like. I'm a population geneticist. Most of my followers on Twitter are also in that or similar fields. People gravitate towards others with similar interests and backgrounds, and so we can exploit that actually to identify kind of network affiliation with labeled white nationalists. So what we did was, so for this individual, say they have 100 followers, 20 of those followers also follow former KKK Grand Wizard David Duke. We give that a homophily fraction of 20%, and we say, okay, this individual is very strongly affiliated with David Duke's network. It's hard to establish a exact cutoff of what's too much affiliation, but in our data we found that homophily fraction of just 2% represents a twofold enrichment of kind of affiliation with those white nationalist networks relative to the vast majority of users that we analyzed. So we used that fraction as our cutoff of defining here's someone who's associated with this community. And so across the 1,800 preprints that we looked at, a few things stand out. So first of all, we found 10% of preprints across all categories have Twitter audiences with more than 5% of that audience coming from these white nationalist networks. These are particularly common in genetics, genomics, neuroscience, but we also see a handful of papers across virtually all other bioarchive categories, animal behavior, even bioinformatics, biophysics, and so on, that appear to be attracting attention from these communities. And in extreme cases, more than half of the tweets referencing a preprint seem to come from these audiences, which I think, again, really speaks to the fact that, at that point, scientists are no longer the ones controlling the conversation about their own work. It's been kind of entirely swept up by groups that are misappropriating and exploiting that work to promote their ideology. And so this kind of leaves us with, what do we do? I think everyone in this panel has kind of addressed that in different ways. Brian's focused on the educational aspects. Joanne is focused on the historical aspects. Graham on the technical scientific, and Brandon on the more philosophical and perhaps polemic side of things. I think I, as the youngest and most chronically online of the group, have focused on more just what do we do with what's right in front of us on social media and other online spaces. And so first, I just want to double down on kind of my own area of expertise. I think there's still a lot of work to be done to simply characterize this problem and adapt information from or adapt strategies from kind of other infodemic research. What are the tools and methods used in kind of tracing the COVID-19 infodemic or climate change or anti-vaccine research? How can that help us kind of understand the shape of this and more concretely study what's going on? I will say, I think in genomics, there's a lot of really unique features in our field that differentiate it from, say, COVID or climate research. If I can get into if anyone is curious in the Q&A. One thing that I think we can all maybe take up in pursuit of this goal is peer accountability. I think in fallout of the Buffalo shooting in particular, there was a Belgian scholar who had an academic affiliation. His name was Michael Woodley. He was cited extensively in the Buffalo Shooters Manifesto, and was essentially suspended from his position at his university for his racist work, as well as I think he was also like into some like cryptozoology stuff, which is weird, but more on that later. Another recent incident, a professor was publishing racial pseudoscience for several years, and was ultimately fired from his tenured position because he blatantly violated a number of ethical guidelines, sharing data from dbGaP and other sources that he did not have permission to share. Essentially, I think there's a strong correlation between violation of just dry ethical norms in science and promotion of pseudoscientific beliefs. And so I think peer accountability is absolutely a way that we can clean house in a sense. I think there's to my point about differences between say the COVID infodemic and this infodemic. I think in something like COVID or climate change, there's a much clearer distinction between say the kind of mainstream scientific consensus and the counter-science or pseudoscience, and that distinction is really what's being exploited by, say, fact-checking strategies or instances where we can lean on kind of the subject matter expertise, whereas in genomics, a lot of the misinformation surrounding the field is I wouldn't even say it's misappropriated, it's just appropriated, where they're taking scientific content and placing it in a different kind of ideological context without even really modifying or distorting the actual scientific content. And I think that's where work that Graham and John November and others are doing on pushing the field to better differentiate what the science actually says from kind of these kind of reified notions is really, really important. So yeah, I guess kind of reiterating that, there's some really great work that Graham and Brian and John and others have done on developing new visualizations. We touch on this a bit in the Nature commentary published recently. And I think this is, it's important to see this as more than just, improving our science. I think doing so inherently, motivates us to better engage with the impacts of our work. If we're anticipating that, a visualization we produce might be misconstrued in some way, that's an opportunity to reflect on why are we doing something the way we are? What are other ways we can represent this information and convey it more clearly and make that more cohesive with kind of what we know and our own kind of moral obligations. Step 3, I think, is this is a fun one. I think a lot of the appeal of weaponized science is not it's not simply a misunderstanding of research. There's an element of subversion and kind of in-group, you get the joke, you understand this secret knowledge, you're part of a community. And I think it, incorporating some of those understandings of the social aspects of how decontextualized scientific information spreads, we can also apply as scientists just in how we're engaging with the world around us. So this is a Facebook page called Dandy Memes for the Dink Darwinian that has just I don't know, dumb, stupid, funny memes about evolution. And I think using that to as an avenue of outreach, particularly to vulnerable young people who are attracted to the opposite end of again subversive information in other realms. I think there's room for us to explore how we can use that in our communication. And finally, I want to reiterate, like I absolutely do not think that this is a problem that we as scientists can solve by doing better science or communicating our science better. I think this absolutely needs to be coupled with broader activism and solidarity with vulnerable communities and anti-racist groups. So I laid this out on a piece I wrote for Science for the People last fall. And I think there's a lot of inspiration we can take from various other scholars throughout history that have taken this up and understood their understood the social context of their research and kind of the necessity of coupling that scholarship with broader social activism. And so with that, I think we can jump right into the panel.
Thank you, Jed. And thank you, everyone. Wow, that Jed, there were comments, and you're going to have to look at the comments in the chat box because you're being referred to as a warrior to go on 4chan like that for the research. That was an excellent talk. Okay, so a second, I have a couple comments. Before I open it up to questions and to the panel Q&A, I just want to point out a couple of things. First of all, I wanted to say thanks to a couple of people. So the SSE Diversity Committee has been thinking about this potential symposium for a long time, and they encouraged me to hold it and convinced me that it was important, and actually did a lot of the legwork to help me decide on ideas for topics and for the speakers and so on. So thank you. I also wanted to let everybody know that SSE is a member of Access Plus. Access Plus is an NSF-funded effort to create a consortium of societies that use the infrastructure of scientific societies to promote change from within in STEM and in our fields. And I have learned a lot from my colleagues on both in the Diversity Committee at SSE and in my Access Plus group. You know who you are. All of this to say that what I hope this means is that our society will continue in the future to continue thinking and talking and having conversations about these things. Brandon put this beautifully to us as an invitation to conversation when he started out his talk, and I think I guess what I'm saying is that I really think this is not a one-off, that our society has to continue with these sorts of activities moving into the future. Okay.